Okay, and these are, as I showed before, these are the infection types that we use. All of these we consider to be incompatibility, including the mesothetic. It's number 4 where we get susceptibility or compatibility, right? In any one of these, when we see any one of these, we know that there is at least one gene for avirulence in the pathogen matching one gene for resistance in the host. That's what the first law of genetics of host pathogen interactions allows us to do by using the gene for gene hypothesis. Getting a little bit off the theme here, but this is one point I'd like to make, and Gordon touched on this in his presentation. I said before, and I showed you this picture where we have all of these, we're saying are low infection types, and this one's the high one here, right? The compatible one, the incompatible, where we have the matching avirulence gene, at least one matching avirulence gene and resistance gene in the hosts here, and this one we don't know what the pathogen or the host genotype is. These genes that we work with, like the SR50 I was talking about, or SR5, they have typical low infection types. So often when we're doing this genetic work, the infection type can give us a bit, so it's not just saying, oh well, it's low or it's high. The low infection type can actually also give us a bit of an indication of what gene might be present. They each have typical phenotypes, and I've just given some examples here. So the Nort infection type is very typical of SR5, the stem rust resistance. The Nort FLEC, typical of SR6. This 1N or FLEC1 infection type we often see on SR21. The 2 infection type where we have the green islands, typical of SR24. The 3 infection type SR29, the X of SR17. There are other genes that also give these infection types, but there are some typical infection types associated with each gene that also allow to add information when we're looking at this genetics, when we're doing this genetics work. And those phenotypes, right, those infection types are illustrated in our book, and you'll get a copy of that in as a PDF with the handouts you're getting. All right, so now going back to this, and this, I suppose if we get, I might just go through this second law and then we might finish for lunch, okay? Yep. So I said there are 2 laws, right? 2 laws of genetics of host pathogen interactions. The first one is this one. The low infection type tells us straight away we've got at least one matching avirulence and resistance gene in the pathogen and the host. The second law, and this is a little bit complicated, but the second law tells us if you've got 2 genes together, 2 resistance genes together that give different infection types, right? Then the infection type, if the infection type of the first gene is lower than the infection type of the second gene, then when you have both of those genes together, you will see low infection type 1. You'll see the lowest one. So if you have a gene that gives a Nort infection type and another gene that gives a 2 infection type, and you have them together in one variety, you will see the lower one of the 2. That's the Nort infection type. All right, so I'll run you through an exercise on that to see how we go, right? So here we have 2 genes for leaf rust resistance. First one is LR1. I have a differential line called TARSA that carries LR1, and LR1 typically gives this Nort or Nort FLEC infection type, like immunity, no real symptoms. LR20, on the other hand, is this necrotic one. You get a small pustule with necrosis around it, right? This is a pathotype that is actually avirulent on LR1 and LR20. It carries both avirulence genes, one matching LR1, one matching LR20. What do you think? So this is our low infection type 1 and our low infection type 2. What do you think would happen if we have a wheat cultivar that carries both of those resistance genes? So this variety Norco was bred in Australia and it carries both of those resistance genes. What do you think this infection type is going to be here? It's a Nort FLEC, right? Now the reason is because this one is lower than this one, so we're going to see that low infection type 1. We're going to see the lowest one of the 2 when we've got both together. Here's another pathogen isolate, another pathotype, a different one. This one carries the corresponding avirulence gene for LR1, but is virulent for LR20. What do you think we're going to get with those two genes now? What infection type will we see with this pathogen isolate? That's right. All right, so what we're seeing here is the effect of LR1. We've rendered LR20 ineffective. It's no longer effective, but we're seeing the effect of LR1, that lower infection type of the two. All right? Pathotype number 3. Virulent for LR1, but it carries that matching avirulence gene for LR20. What is it going to do? We're going to see LR20 this time, aren't we? So here we've knocked out the effect of LR1, right? But we're seeing this LR20. All right, so you could take 3 pathotypes and inoculate Norca, and depending which one you use, you'll see a different infection type. That tells us that Norca carries more than one resistance gene because these infection types are different. And if we know the pathogenicity of the isolates on those genes, then we can deduce a genotype for Norca using the gene for gene hypothesis. What about this one? Doesn't carry the matching avirulence gene for LR1, does not carry the matching avirulence gene for LR20. Virulent on both genes. All right? It's going to be virulent. It's basically invisible because it doesn't carry the avirulence gene for LR1, it does not carry the avirulence gene for LR20. So it's invisible, the traffic light is green, the pathogen can grow. All right, one more trick for you. Oops, sorry, one more for you. And this is based on a true story. So back in 2002, I got a leaf rust sample sent to me from Victoria, and I put it on my differential set. And my leaf rust differential set, incidentally, has all 3 of these, right? So I took the notes. I'm working my way through the differential set. I see Tarsus giving me a 0. I see Thews giving me a 4. And I'm thinking of the gene for gene hypothesis. So I'm thinking to myself, this is what I should see, right? Before I even got to Norca, I'm working my way through the set and I'm thinking, okay, Norca's going to be a 0 fleck. And it was that. Straight away I thought there is something wrong here. The first thing I thought is we've made a mistake with how we've planted the differential set. That's the obvious one. Someone had put the wrong seed in the wrong place in the pot, and this was not Norca, but in fact we repeated the experiment and it was correct. Very hard to explain, right? But there was, and it took me, I had 25 years of experience working with leaf rust when I saw this, and it took me about a month to work it out. But what it told me was that Norca carries a second uncharacterised Thew. Let me rephrase that. Tarsa carries a second uncharacterised resistance gene for leaf rust resistance that was giving a very similar infection type to LR1, right? So Tarsa actually carries not just LR1, but another gene that we didn't know about. We hadn't seen it before. And it turns out that that gene is also present in a line we call Morocco. I don't know if any of you heard of Morocco wheat? Morocco is a famous wheat genotype in the rust world because it is susceptible to everything. Well, I actually found a resistance gene in Morocco, and it's the same gene that's present in Tarsa. We didn't know about its existence until we found this particular rust isolate here. All right? So Tarsa carries LR1 plus another gene we now call LR73, which is also present in Morocco. Norca, however, only carries LR1 and LR20, and this pathotype is actually virulent for both of those genes, but avirulent for this second gene in Tarsa, this LR73 gene. A little bit complicated, but the point I'm trying to make here is that this whole idea, this second law of host pathogen genetics, where when you have 2 infection types and one is lower than the other, right, that you always see the lower one, allows you to actually do some genetics on fixed stocks. I didn't have to make any crosses. I didn't have to do what Mendel did. I didn't have to do what Flor did. I mean, ultimately I did have to do this to prove it, but I was able to form a very accurate hypothesis just using the gene for gene hypothesis to work out what was going on here without making any crosses, just by my knowledge of the genes and my knowledge of the resistance genes and my knowledge of the avirulence genes and the pathogen. All right? So the second half of the lecture, which we won't go into yet, we'll break for lunch, but the second half of the lecture, what I'm going to talk about is using the gene for gene hypothesis in resistance breeding. I mentioned this at the outset. I said it forms the basis pretty much of all of the work we do in resistance breeding. We apply the gene for gene hypothesis in two ways. Firstly, in race analysis, and secondly, in gene discovery, when we're looking for new rust resistance genes. We use the gene for gene hypothesis in both of those cases. And I'll go into each one of those after lunch if time permits. All right, so let's grab some lunch and...
So we've got, after lunch, we've got a half-hour session of question and answer. So we'll have time for Robert to finish his presentation, and then we'll start on the country reports. So before we break for lunch, we should probably get an update on the cricket score.
Hold on. 133.
Whoa.
You get 98% chances of it.
The probability of winning South Africa.
But there's a confounding factor. There was a rainstorm,
Oh, could end up in it.
so that changes things a bit.
Okay. Okay, so we'll see you back here in 1 hour.
No, it was a funny storm or something.
Dr. Gordon, sir.
Yeah.
So, the paper you referred to that you're going to give them is the one